Hello, and welcome to our listeners around the world. You're listening to JAMAevidence, our podcast series based on core issues in evidence-based medicine. Today, we're discussing early stopping in clinical trials. I'm your host, Dr. Roger Lewis, Senior Statistical Editor for JAMA and editor of the JAMA Guide to Statistics and Methods series. I'm here with Kert Viele. Kert, can you go ahead and introduce yourself to our audience?
Hi, I'm Kert Viele. As you said, I'm a biostatistician and the Director of Research at Berry Consultants.
Thank you so much for joining us today. I should let our listeners know that I am also a member of Berry Consultants, and Kert and I have worked together on many projects. So your article talks about clinical trials that stop early. Let's start with the motivation for even considering this topic. What are some of the reasons that clinical trials might stop early?
Usually there's a couple reasons you would want to do this. When we plan for a trial, we plan for a sample size that's large enough to answer the question, whatever that research question might be. And what often happens is when we actually run the trial, we can get an answer to our question earlier than that planned sample size. And that answer might be negative, might be the drug therapy doesn't work. It might be positive that it does. And so when we get that answer earlier, it's valuable to go ahead and get that answer to patients, and it's valuable to get that answer to the scientific community so that we can use our resources to answer other questions.
If we might get an answer before we reach the maximum sample size, how do we know when that happens?
So usually what we do is we make planned interim analyses. So we decide if the full sample size is, say, 300. We say that we're going to look periodically at the data. So maybe at 150, 200, 250. And we have predefined rules which say if these conditions are met, often a p-value being low or something similar to that. If those happen, then we might stop for success. Similarly, for futility, if the p-value is very high, or if the chance that the trial may ultimately be successful is very low. But essentially, we're very sure the drug works, therapy works, or we're very sure that it doesn't, is where we would do this at these interim analyses.
So when you say we look at the data, we're often taught that it's very important to remain blinded to data as it's being accumulated to avoid bias in the study, bias in the sense that investigators alter either the types of patients they enroll or the way they assess outcomes based on knowledge. So who actually looks at the data, and how do we avoid that kind of bias?
We is the broader we here. Usually these roles get divided up. So the person running the trial, making decisions about the trial, seeing patients, they do need to remain blinded and unaware of the accumulating data, as you said. So usually what happens is when I say we, this is usually a group of people. They're firewalled from the main study. So basically no one at the trial would know what the results are, unless, of course, the trial stops. Then it would have to be communicated. But this is done by an independent third party.
Now, you mentioned stopping both for early success, where you have conclusive evidence of benefit, or for futility. So let's focus on these separately. So for early stopping for success, what are the things we need to do to make sure that we maintain the statistical or inferential integrity of the trial?
The central thing that we worry about in this case is because we're looking at the data multiple times, we have to be careful that whenever you do something multiple times, you have a multiple chances of making a mistake. If the therapy actually doesn't work, we don't want to falsely claim it does. And if we look at the data over and over and over again, and we do something like a rule p less than 0.05, 0.025, one-sided or two-sided, doing that multiple times, we can increase our chance of making a false claim of efficacy because of just repeated looks. In that case, what we do to correct that is instead of looking at 0.05 or 0.025, we would look at a lower level. Maybe it's 0.001, maybe it's 0.022. There are mathematical formulas which say how we need to make those more stringent thresholds to make sure that we don't make a false claim of efficacy.
So if we use more stringent thresholds before we claim that a given data set is positive, doesn't that run the risk of lowering our power in the trial?
There are a couple solutions to this. It will, if you keep the maximal sample size the same, usually you do have a slight reduction in power. It's often a very small number of percent, say 1%, 2% by doing this. And you view that as potentially offset by the sample size savings. The other thing that you could do is increase the maximum size of the trial. So instead of running a 300 patient trial, maybe the maximal sample size is 320, but I can still stop earlier than that 320. So what happens in those cases is on average, I'll get a shorter trial, but it's always possible I may end up needing those extra few subjects. So that can go either way.
So when you say a sample size savings, it isn't a guaranteed sample size savings because you have to be able to go to 320. It's just on average, you might save some sample size. Is that fair?
Yeah, that's fair. Usually in these cases, and it obviously depends on the details of your study, but a lot of times these studies maybe have a 20 to 30% chance of needing to go to the maximal sample size, sometimes less, sometimes a little more. But the chance of needing the maximum is usually pretty limited.
So I know there are specific names given to these strategies or plans for allocating the risk of a false positive or type 1 error across the interim analyses. What is the most common strategy and how does it spread that risk?
So the most common strategy you'll see is something called an O'Brien-Fleming boundary. And what's important about that kind of boundary is the early looks when you're, say, looking at halfway through the data set are very, very stringent. You need a very low p-value, very strong evidence in order to stop. And as you move towards the ends of the trial, towards that maximal sample size, the p-value starts to get very close to 0.025, which you need. It might be 0.022, somewhere in that range. So the idea is that you need really strong evidence to stop early, and you basically at the end of the trial end up with a test that looks similar, not exactly equal, but similar to that final test. So that's the most common one. The advantage to O'Brien-Fleming is it has a very, very minimal impact on power. You don't lose much, your last question, but you tend to save a decent amount of sample size on average by doing this.
So if the advantage is the possibility of stopping early if a treatment turns out to be truly effective, what are some of the downsides of this approach?
The construction of the method is, and I'll start off with the positive, something we shouldn't worry about. If you stop early, we've protected ourselves from a false claim of efficacy. So when we stop early, we can be confident the therapy works. The downside is that we also would like to estimate how well does it work. So do we have a strong effect or not? So going back to what we just talked about on the O'Brien-Fleming, if you need very, very strong evidence to stop at an interim analysis, you may need a very, very strong effect. And whenever you're only looking for strong effects, it's possible that those strong effects are overstated. So you could run into a situation where the therapy, say you get a hazard ratio of 0.6, you have to look at that and go, I was looking for really strong effects. Maybe it's not quite that good. So there's always this thought that maybe there's a little bit of bias attached to this. And so that's what people will worry about in these settings.
What kind of bias, sort of quantitatively, should we be thinking might be associated with results from trials that stop early?
So that depends on the setting. That's a loaded question. There's a lot of room to go at this. My personal thought is that in many cases, if you've designed this well, the bias is fairly minimal. So maybe it's on the order of 10% overstatement of the effect. I would tend to go, hey, it's probably a little high, but not too much. One thing I think you want to be really careful about when you design these studies is, suppose I'm talking about something that changes blood pressure, and I know that I'm never going to change it by more than 10 points. I could set up a study that looked way too early, and so you need a 30-point change in blood pressure to stop. If I suddenly get that 30, 35 point change in blood pressure, I'm not going to believe it because it's implausible. So you want to be really careful setting up these designs so that when it stops at a plausible value.
So when you mentioned the two general categories of reasons that a trial might stop early, you mentioned both stopping for efficacy, which we've discussed, and stopping for futility. So what is futility and what are the considerations in choosing whether to stop for futility?
So futility would, of course, mean that you're concluding that you're going to go ahead and stop the trial, stop investigating the therapy. You might conclude that because you have evidence that the therapy doesn't work or is harmful. You can also do that if you feel that your current trial has no hope of demonstrating efficacy. So you're at the point you have some modest effect, but I'm not going to get what I need in order to win, and thus I shouldn't be expending resources in a futile effort to demonstrate efficacy. By far, futility is the more valuable way to stop early. Lots of therapies don't work. We can usually stop for futility earlier than success. We get bigger savings. Every trial should have some kind of futility rule.
One of the questions I've often heard comes up when investigators have groups in place monitoring the safety of incoming data, and they want to know what to do if those groups notice an unexpected positive result while they're looking at safety. Should they just go ahead and stop the trial at that point, or what should they do?
So you want to be really careful in this situation. In order to control the probability of making a false claim of efficacy, you need all of your looks to be pre-planned. So you need a protocol written in advance that says, under these conditions, I will stop. And because of the math, because of the way we're controlling our chance of a false claim of efficacy, we can't look at the trial in any kind of ad hoc fashion. So if you are concerned that that's going to be a possibility, maybe at a safety look you might be doing it, you either need to include those chances in advance. So you could say at these points, if the data is sufficiently strong, I'll stop early and just include those in your calculations. Or you have to accept that I can't stop for efficacy at these points.
When we discuss stopping early for efficacy as opposed to futility, we pay a lot of attention to the risk of inflating the type 1 or false positive rate. What are the considerations on the possibility of getting a false positive when we consider stopping for futility?
So essentially, there are none. We're talking about whether we make a false positive. And so the key part of that is positive. If we're stopping for futility, we're making a negative decision. Given that there's no chance to make a positive decision, we don't have to worry about inflating our chance of a false positive. We're just not making a positive. So there's no alpha penalty. You don't have to worry about those looks. There are some thoughts that there's a chance you may look for futility and recognize, oh, the data looks fantastic, and so I'm going to go ahead and stop the trial. I usually recommend not stopping in an ad hoc fashion, so I don't worry about this. But generally, futility, you don't have to pay any kind of penalty. There's no alpha penalty. There's no false positive multiplicity concerns.
The article that you wrote for JAMA used as its motivation a study of a treatment for glioblastoma. Can you go ahead and tell us a little bit about what those investigators did and what they found?
So in this article, they were investigating temozolomide plus tumor treating fields as opposed to temozolomide alone. And they were looking at what resulted in higher survival. So the hope was that adding tumor treating fields would increase survival. They included interim analyses at periodic times, all pre-planned, which said they would look at the data, and if the results were good enough, they would stop early and claim efficacy. And so what they did is they continued the trial. They did these looks. When they got to 315 patients, they found a p-value of 0.001, a hazard ratio of about 0.62 in this study. And so the 0.001, that was lower than their pre-specified threshold. And so that gave them the option to stop early and claim efficacy in this case.
So, are there any additional things readers should think about when they're looking at results from a clinical trial that stopped early?
So there's always endpoints outside of efficacy. That's typically of extreme importance. Safety is very important. You should consider this when you're designing a trial to make sure that there's enough evidence for safety. You want to make sure that when the trial stopped early, it didn't stop so early it left safety questions unanswered. You want to make sure the trial's pre-specified, the data quality is good. All of these things that go into a good, robust study are going to come into play again. But typically, the most important thing is to have a sufficient safety database to go along with this early stop on efficacy.
So, what are the key takeaways for our audience regarding clinical trials that stop early?
First, I think you should always have the ability to stop for futility. It is often the most efficient thing that you can do. If a therapy isn't working, we should make sure that we move on to the next thing. The second thing that I would consider is that you always want to make sure you're controlling for false claims of efficacy. So you need a pre-specified plan for when you're going to look at the data and what the rules are going to be for stopping early. You want to make sure that you follow that plan. Don't do anything ad hoc. Third, you want to be careful for potential biases in your estimates. So you want to plan your study. Don't stop at implausibly high estimates. So you may want to just run your interim analyses later if you're worried about getting into that situation. And fourth, all of the things that go into a good trial, making sure that you've demonstrated efficacy and safety, you've run the trial well, you have high quality data, all of that still applies here, and if not more so, because you're making sure that you're looking at the data repeatedly, so you want to make sure you make no mistakes.
I'm Roger Lewis, and I've been speaking with Kert Viele about clinical trials that stop early. You can find a link to his article in this episode's description. This episode was produced by Shelley Steffens at the JAMA Network. To follow this and other JAMA Network podcasts, please visit us online at jamanetworkaudio.com or search for JAMA Network wherever you get your podcasts. Thanks for listening.
This content is protected by copyright by the American Medical Association with all rights reserved, including those for text and data mining, AI training, and similar technologies.